Okay, so we're going to start talking about cellular respiration. And the first step of cellular respiration is a series of chemical reactions that are called glycolysis. And glycolysis is something that every single organism on the planet is going to do. Whether you're talking about a bacterium or a yeast cell or your cat or you or an oak tree or a coral reef or anything, all of those organisms are going to do glycolysis. The very first cells on the planet, the origin of life, did glycolysis. And so it's only a few steps, and we're only going to kind of look at an overview summary of the key reactions of all of these things. And so for us, it's going to be very simple, but it's one of at least a 4 to 5 part process depending on how you look at it. And so if we talk about respiration in general, this is our equation for cellular respiration. We're going to start with glucose and oxygen, and we're going to end with carbon dioxide and water. But that's all of respiration. In this first part of respiration called glycolysis, we're going to start with this molecule of glucose. I'm just going to say glucose, but it's the same thing, the C6H12O6. So we're going to start with the glucose, and we're going to use up 2 molecules of ATP. And we're going to produce 2 molecules of something called pyruvate. And we're going to also get out 4 molecules of ATP. We're also going to make one other sort of a molecule, which is called NADH. And that is what glycolysis is going to be. Well, if you look at what's going on here, we start with glucose. That's our sugar. We end with these 2 molecules of pyruvate. Pyruvate is also a sugar. Well, pyruvate, it's the same thing, right? So we have 2 of the exact same molecule. If glucose has got 6 carbons and pyruvates are exactly the same, and there's 2 of them, you can probably guess that each of these pyruvate has got 3 carbons, because 6 divided by 2 is 3. So pyruvates have got 3 carbons. We're going to get 4 ATP, but 4 ATP aren't really our paycheck. So why don't you think the 4 ATP are what our paycheck is going to be? And the answer is because we used up 2 in order to get to this point. So we use up 2, we get 4, we're going to net 2 ATP. And then this other molecule of NADH, that is something special. That's carrying an electron. And we're going to learn that these molecules of NADH and another one called FADH are actually far more important to us than this one-off reaction where we get an ATP. So as we go through all of these reactions, whenever we talk about what it is we're kind of conceptually doing in every step of cellular respiration, you are starting with this molecule of glucose. Glucose isn't super unstable, but because it is a larger molecule, it has a lot more energy that it can but it can make available to the system than a smaller molecule. And so for every step of respiration, we're chipping away at this glucose molecule, and we're making it smaller and smaller and smaller. We're breaking it into smaller parts. And every time we break it into smaller parts, we're going to a smaller molecule which is more stable than the bigger molecule that we began with. So if it's progressively becoming more and more stable, that means it's progressively losing free energy. So if we go back to these sorts of graphs, these ones that we had looked at last unit with about free energy, we're talking about Gibbs free energy, or whatever kind of measurement you want to have and how they change over time, we had exergonic reactions. And exergonic reactions begin with a large amount of free energy and then end with a lower amount of free energy. And that's because this molecule has a lot of free energy because it is unstable. This molecule has less free energy because it is more stable. So every time that we do one of these reactions, whenever we break these bonds and go to a smaller molecule, the reactants have more free energy than the products. And so because there is less free energy available inside of that molecule, that means that energy was released to the system. And that energy that has become available can be used to drive endergonic reactions, the ones that go uphill and let us make new molecules. And so those molecules that we can make, they're going to be kind of one of 2 flavors. The first is maybe we're going to make molecules of ATP. ATP we can use for energy. We can use it elsewhere in the cell to drive other sort of reactions. And so we can couple the formation of ATP with these exergonic reactions where we're breaking bonds. The other thing we could do is when we break that bond, remember that in all of these molecules it's a carbon bound to a carbon, right? Because all sugars have got a carbon backbone. Well, all of these carbons are joined together by a covalent bond, which involves the sharing of electrons. Well, when we break that bond, that means that this electron, which is a high energy electron, a very special kind of electron, it's high energy, it's moving at a very rapid pace, it has a lot of kinetic energy available, it is far out within the electron cloud. We can now take it to another location to do a different set of reactions that we're going to see later. And so we can take that electron that used to be within the bond between these two carbons to another part of the cell to do work. And when we remove the electron, when it involves the transfer of an electron from one atom to another atom, that is going to be called a redox reaction. A redox reaction. And a redox reaction involves this transfer of an electron, and the molecules that are going to carry those electrons are either NADH or FADH2. Both of those molecules are used to carry an electron. You'll notice they're kind of big. They got like, this NAD stands for a larger molecule. It's a big old molecule, and it's just because since it's a large molecule, it can absorb the punch of a high energy electron and take it somewhere else inside of the cell. So in glycolysis, we're going to see the formation of pyruvates, ATPs, and NADHs. Every single one of these reactions is always going to be exergonic, which means energy is going to become available, and that energy can be used to either create ATP or perform some sort of redox reaction where we carry a high energy electron to another part of the cell. So now we can actually talk about how we're going to do that. So when I draw these reactions, I usually use little circles to indicate the carbons. I think that makes it a little easier to follow inside of each molecule. And since it's the first reaction, we're starting with our overall reactant, which is glucose. That has 6 carbons. So I got 6 circles to represent the 6 carbons of glucose. And we are going to add ATP into this chemical reaction. And the reason we're doing that is we're going to transfer one of these phosphate groups from the ATP onto this sugar. So we've still got those 6 carbons of the glucose, but now they've got a phosphate group attached to them. Now we're going to do it again to add another phosphate group. And we can't add the phosphate groups to like the middle of the molecule, so they get added to the tips. But remember that phosphate groups are one of 6 functional groups, and functional groups add instability to a molecule. And so we've just added 2 of the most unstable functional groups to this already energetic molecule. So we've just given it a situation in which it has a ton of free energy. And it has a ton of free energy because it is very unstable. So it wants to become more stable, which means it wants to do some sort of exergonic reaction. That way, it can have more stable bonds, there's free energy available, and it wants to basically break in half. It probably would be happy breaking anywhere, but the reason why, in our case, we would like to do break in half is because if it breaks in half, we can form 2 molecules that are the same. That way we can do all subsequent reactions the same on the 2 different molecules. If instead it broke at like one, carbon number 1, and then these 5 carbons, and it had a 1 and a 5, now we got to do a whole new set of pathways for those 2 different things. So it's easiest if it breaks right in the middle, then we've got 2 that are the same, and that's what we're going to see happening. We're going to get a few different reactions that are going to occur where the phosphate groups are going to be lost, but ultimately we're going to end up with 2 molecules of a 3-carbon sugar called pyruvate. Well, we just went from this 6-carbon molecule to 2 3-carbon molecules. Right before that happened, we had this very unstable molecule. Now we have one that is more stable. We did all of the things we're describing over here. We had an exergonic reaction. Well, when we have one of these exergonic reactions, or whenever you see one of those bonds being broke, every time you see one of those little circles come off, you know that energy became available. So you must have created something. Otherwise, you wouldn't have done it. In this case, we're going to use the energy of this exergonic reaction to drive the formation of 4 molecules of ATP. So we're going to get 4 molecules of ATP. And also, since we broke that bond, that means that one of those electrons could have been pulled away and used somewhere else in the cell. And so we're also going to create molecules of NADH. And so ATP can just be used for energy right away. The NADHs are going to be carrying a high energy electron that used to be within this carbohydrate molecule, this molecule of glucose. And so those are our products. We've got 4 ATP, NADH, 2 molecules of pyruvate. We invested 2 ATP, so we net 2 ATP as our yield. We began with glucose, we end with pyruvate. Glucose has got 6 carbons, pyruvate has got 3 carbons.